thermophiles. So what thermophiles? We all know that comes from the Greek word thermos, meaning heat, and the philos meaning the loving. So as the name suggests, thermophiles are the heat-loving microorganisms that not only tolerate but actually require high temperature for their growth and the survival. And within the range of the 45 to the 120 degrees centigrade. So they are often found in the extreme environment like the hot spring, deep sea hypothermal vents, and even the man-made habitats like the compost. So thermophiles, now the question that why you are going to means why you are means concentrate or going to discuss this topic, because we all know that the thermophiles produce enzymes. And those enzymes are known as the thermozymes, so that are naturally stable and they activate the high temperature. So these enzymes do not denature easily, and this means characteristics, or you can say the property, make them the ideal candidates for various industrial applications where the high temperature are part of the process. So let me highlight a few major applications of the thermophiles. In molecular biology and the genetic engineering, we know that the Taq polymerase that isolated from the Thermus aquaticus, and this enzyme is the backbone of the PCR. Then the application of the thermozymes in the biofilms and the biomass conversion, food and dairy technology. The thermophilic bacteria like the lactobacillus thermophilus are widely used in the yogurt and the cheese production. There is also applications of thermophiles in the pharmaceutical and the chemical industry, detergent and the cleaning agents, and most importantly in the environmental bioremediation. So in the environmental bioremediation, that they have the ability to degrade the toxic compounds and the heavy metals even under the harsh conditions. So they offer the eco-friendly solution to clean up the industrial waste and the oil spills. So beyond the industrial values, the thermophiles also expand our understanding of life's adaptability. So now in the current research means there is the implications in the astrobiology, where the scientists are or the researchers are investigating the possibility of life in the extreme environments on other planets. So in short, just I would like to conclude that thermophiles are not just the survivors, they are enablers of the innovation, and they challenge our notion of the limit of life and offer the tools that make many modern technology faster and more efficient. So thank you everyone for joining this roundtable discussion session. So, and I look forward to an insightful and the engaging sessions ahead. So I now request all the who have joined this session to unmute yourself and kindly share your research experience so that each of us can learn from each other.
Yeah, before that, I will request
Yes, sir.
the participants who have not introduced.
Sure, sir.
For example, Sindhu, Srinivas.
Sindhus and
Nenavat, Padma.
Nenabat.
Please introduce yourself.
And also write your affiliation and designation in the chat box.
Sindhu, please introduce yourself. Srinivas, please introduce yourself.
Hello, sir. Good evening, sir. This is Srinivas, first MJC from Dr. BRR Government Degree College, and my supervisor by Dr. A. Madhuri Madam from Mahabudnagar district, Lechela College. So I want to discuss about thermophiles.
Very good.
So
Very good. Vipran sir will give you opportunity. Don't worry about it. Okay?
Okay, sir. Thank you.
Then Kumari Pallavi. Kumari Pallavi, Fiza, Nenavat Padma. Please introduce yourself. Earlier, don't waste the time. Fiza.
Good evening, sir. I am Pallavi. and I'm student of BSc Biotechnology second year and pursuing it at Gautam Budh University, Greater Noida.
Okay. No.
Thank
Good, good, good. Then Aisha, Brian D'Souza. Be fast. Don't waste the time. Snehal Punde, Prafulla Patil. Introduce yourself in one or two lines so that we'll go for discussion. Why you are joining the if you don't want to introduce yourself? Either write the message in the chat box. If your mic is not working, yes, D'Souza from Mumbai. Okay. Yes, Bipran sir, now you opportunity to one by one.
Yes, sir. So, my dear participants, if anyone want to share his or her research experience, now this platform is open for the discussion.
Sir, can I share?
Sure, sure, Srinivas.
So thermophiles means thermophiles are microorganisms, mostly bacteria and archaeas that thrives at a high temperature, typically between 45 degrees Celsius to 80 degrees Celsius. They are at present. Their habitat it's found in hot spring geysers and volcanoes, deep seas hydrothermal vents and composite hips. So, in our example, thermos aquatic used in PCR for DNA application, sulfolobus and pyrococcus furiosus. And they are thermophiles are enzymes. They produce heat stable enzymes such as tachypolymerase useful in biotechnology and industries. There are types moderate thermophiles of 45 degrees to Celsius to 60 degrees Celsius. Extreme thermophiles, hyperthermophiles are 80 degrees Celsius to 110 degrees Celsius. They are in live adaptation, especially proteins and cells membrane that don't denature at a high temperature. DNA binding proteins that protect genetic materials. Important uses used in industrial process like biofuels, detergents, food processing, useful in scientific research, especially in molecular biology. Sir, this is my brief about thermophilics, sir.
Thank you, Srinivas, for sharing your means. So, are you pursuing any research means in thermophiles?
Hi, yes, sir.
A little bit, if you can share your research.
Sir.
So, it's I request you to little bit share your own research so that we can learn from you.
Hi, yes, sir. I'm learned from this, my research.
Okay, thank you. So now we would like to hear from the Dr. Halami sir. Am I audible to you, sir? I think that's some network issue. So any one participants, kindly, if you have any research experience or any comment on the thermophiles, you may unmute yourself. Please share your.
Hello, doctor.
Yeah, hello, Dr. Majid.
Uh, see, this one first because these thermophiles are having a lot of benefits as well as we can do a lot of research in many streams. So better we focus on one stream, either in the food or pharmaceuticals or environment. As far as about my research, I have worked on several domains means in microbiology, especially
Hmm.
environmental and pharmaceutical microbiology. So in one of the research, I worked with the microbial fuel cell. So in microbial fuel cell, we have isolated the organism, geobacter organisms, and simultaneously we were treating the uh, wastewater, brewery wastewater, and generating the bioelectricity. So that has published in one of the elsewhere journals. So here, what we can do is we can use these thermophilic organisms for generating the organic waste at high temperature. So generating electricity from organic waste at a high temperature, this we can do, I think. And because I did not work with the thermophile, but I worked with the psychrophilic organisms. But my intention was during my research, I was interested to work with thermophiles. So these are my views. And also this will help in carbon capture. So this will
Yes.
also be if we go for the engineer if we uh, engineer these thermophiles to fix the carbon dioxide, so that will definitely help the climate, right?
Yes, sir.
Yeah.
So thank you for sharing your research insights, sir. And this will open up the new perspectives in this discussion. So any participants kindly share your research means experience, or if you want to put any comment on the thermophile, you want to share your any experience on the thermophiles, you can unmute yourself. Sneha Punde, Sachi Singh, Praful Patil, Kumari Pallavi, Fija, Brand D'Souza.
Dr. Loknath has joined. Dr. Loknath. And one more fellow just told that he is working on thermophiles.
Dr. Loknath sir is joining.
Hello, doctor.
Hello.
Yeah. Hope you can hear me. I have been working on this thermophiles for treatment of petroleum industrial wastewater, and using these thermophiles, we had been involved in biodegradation of petroleum hydrocarbons such as low molecular weight, high molecular weight. Also, this thermophiles is we collected these thermophiles almost 8,000 feet in depth from Saudi Aramco. It is the petroleum industry in Saudi Arabia, and it withstands up to 80 degree, and it was able to degrade the petroleum hydrocarbons. And also we try to collaborate with the industrial sector, and we try to use this in the secondary treatment process of refinery wastewater, since Saudi is having a high temperature ranged from 45 to 55 degree, where the normal bacteria cannot survive and cannot be employed in the secondary treatment process. So we try to implement this process. And apart from that, there are also some halothermophiles has been identified, which can survive both in halophilic, that is on saline condition, as well as on thermophilic condition. Because mostly in refinery wastewater, the salinity will be high, and as well as the temperature is also high. So in order to balance both and withstand the stress condition, this type of halothermophiles can be employed. And we have identified certain bacterial strains such as Acrobacterium, Bacillus, Stenotrophomonas, and then Pseudomonas. Some of these kind of bacteria has been already identified, and we have also published several things. And also we are in consultancy work with those petroleum industries. This is a kind of experience we are having with the thermophiles.
Okay.
Great, doctor. But I have a bit doubt. This one, you are working on this thermophiles now for the petroleum. What exactly means you are doing? You are isolating the organism, and then what are the applications for
Yeah,
isolating?
we try to enrich the consortium from the
Okay.
where the petroleum mud is there, where they are digging
Mm-hmm.
this, exploring this, because we want thermophiles from those conditions, as
Mm-hmm.
previously mentioned by some of the researchers that they collect it from volcanic environment and other hot springs. So we try
Yes.
to go into this field, real field, and we try to get this mud 8,000 feet, something like that. So we try to get those samples.
Means for a bioremediation, right?
Yes, exactly.
Okay,
Yeah,
okay, okay, okay. Great, great. Yeah.
Mm.
okay,
Oh.
sure. And apart from that, there are many applications. Also, biosurfactant has been produced by those bacteria, and it can be applied in many of the aspects, even in phenol industrial wastewater treatment. As
Mm-hmm.
the previous professor said, he is working on microbial fuel cells.
Mm-hmm.
Yes.
Yeah, it can be also employed, but still
Mm-hmm.
there is one more thing is oxygen. Because if you
Mm-hmm.
put it at a high temperature, the wastewater will start evaporating.
Okay, yeah. Mm.
So there is some sort of challenges also in handling these thermophiles.
Okay, okay. What about if we go for with the genetic engineering? Is it possible with that? Uh,
Yes, of course. Why not? Because we have to enumerate what kind of gene is representing this kind of activities.
mm,
So we can work on that, and then
mm.
we can try to isolate those things and go for the sequencing or library. So there are many aspects to work on that. That is a completely separate area. I think
Yes.
many youngsters can work on that, and then they can explore to the scientific community.
Even extracting the enzymes which can work instead of using the whole organism, so we can extract or we can produce the thermophilic enzymes and use in this the bioremediation process.
Yes, yes, of course.
Yeah, yeah. Okay.
Thank you very much, doctor, for the opportunity, and thank you for the
Dr.
team.
Loknath is there. Loknath.
So thanks for presenting
Please.
your findings so clearly, sir. So it was a thought-provoking and enriched the overall discussion. Thank you so much, sir, for sharing your research. Now,
Dr. Prakash Halami is there.
yes,
Dr. Loknath is there. Please.
Dr. Loknath, sir. Am I audible to you, sir? Dr. Loknath Deshmukh, sir.
And Dr. Prakash Halami is
It's,
also
it's
here.
only audible.
Okay.
Yes, yes,
Good
you are
morning.
audible. You are audible, audible.
Good evening there. Yeah, let us take the opinion of other experts and then let us get that feedback from their side. Of course, I am there otherwise to explain the stories of the thermophiles and any other extremophiles and all other methanogens and other microorganisms.
We would like to hear from you,
Please go
sir.
ahead. Please go ahead, Tiwari sir.
Prakash Halami, sir, please introduce.
Okay, I
Introduce
am
yourself.
Dr. Prakash Alami from CFTRI Mysore. I am presently at US on DSR ICMR International Fellowship at Virginia Commonwealth University in the Department of Pharmacology and Toxicology. I am basically studying here the gut microflora associated with the gut permeability and the leaky gut. So again, the full microbiology, how microorganisms actually interact with the host and their beneficial properties. So this is what actually which I am studying here. And then I will be here till this month end. Then I am back to India in August. Thank you.
Thank you, sir. Dr. Loknath, sir.
चांगले.
Hello everyone. Hi sir.
Hi. You're audible, sir.
Hi. Very good evening. Very good evening to everyone. I'm Dr. Loknath Deshmukh. from Chhattisgarh. I'm currently serving as assistant professor in ITM University Raipur, and I'm very junior among you all. You all are very senior, and it's a pleasure to join here and listen you all. Actually, I'm just learning here, and basically, I was technical issues by connecting this meeting. So I had something like methonogens के बारे में मैंने सुना था, although basically my domain is mycology. So time I was trying to explore some fungi. They are under the thermophiles. So I'm just getting the information from you all. It will be great.
Thank you, sir.
Okay, okay. Now, let me tell again, let me make one comment. As many of my students work for MSc dissertation on the thermophiles, and the major problem these students were facing is during isolation, the agar media get evaporated. That was the major hindrance for the isolation of thermophiles. So, the cultivation can be in liquid media, but isolation on agar media is a very, very difficult for all these thermophiles. And my PhD students, they worked on thermophilic actinomycetes. They play a very important role in soil, particularly during summer season, when other organisms die, because when the soil we consider live if the organisms are there. And during summer, because in many parts of the world, the sunshine is so high, temperature increases so high, many microorganisms get death occurs either because of the higher temperature or because of the evaporation of water. At that time, the actinomycetes are playing very important role. They survive in the soil at high temperature and maintain the soil fertility. That study has been carried out my PhD student and MSc dissertation students. Yes, you can proceed now, Bipran sir.
Yes. So Aisha, Arvind Kumar, Kumar Pallavi, Praful Patil, Yashpal. Anyone, if you can just put a comment on the topic, that why you have joined this session, what's your objective, that you can also discuss here. If you have any plan to, means any research on the thermophilic, that we can discuss here.
Sir, I am Yashpal. I joined the meeting because I have always been fascinated by microbes. And what
Great.
what is the mechanism which prevents these thermophiles, means which help these thermophiles to survive such high temperature? I believe it's a miracle of a nature.
Yes,
And
sir, please introduce yourself.
sir, I am assistant professor in Chaudhary Devilal University, Sirsa in Haryana, in field of biotechnology.
Very good, very good, very good.
Yeah.
So
I
please
was reading.
continue.
Yeah, I was few days ago. I was reading that microbes have been found even in very upper strata of our atmosphere. Also, they are beyond in the ocean. Also, everywhere they are so cosmopolitan. So there is something in their genes, I believe, and that genes, if we can, genetic engineering, get those genes in somewhere. Like for example, in PCR, we are getting that Thermus aquaticus gene, Thermus aquaticus polymerase we are using. So such thermophilic organism, if we can isolate their genes and we can use in our some other industry, it can really give us rich dividends. This is my
Yes,
thinking.
what you are talking about the genes. I will tell you one of professor in Maharashtra. He was working
Yeah.
on transfer of thermophilic gene in rhizobium and azotobacter.
Okay.
Why? Because again, as I have told you, the problem biofertilizer will die during summer season.
Sir, actually, I have got some urgent work. I'll rejoin in 5 minutes back.
Okay,
I got
okay,
urgent call.
okay, okay. So I think okay. So everyone is mute. So one of my friend was working in transfer of thermophilic gene in the microbial inoculants. Why? Because the microbial inoculants, if they are transferred in, thermophilic gene, if it is transferred in microbial inoculant, they will work during the summer season. And in India, it is a major problem. There is a 3 month summer in India. It is generally said that if we add microbial inoculant in soil, they will survive for 3 years. There is no need to add every year microbial inoculant. But because of the summer season, there is always danger that they will die. The same is for azolla. Azolla survive, it is an aquatic fern. The problem with the azolla is that if there is a direct sunlight to the azolla, the growth is stunted. That is a limitation. So by transfer of thermophilic gene in the future, we may overcome such type of limitations. Yes, sir.
Thank you, sir.
Yes. Any other again want to make a comment or have studied for MSc also? They can contribute their experience.
Doctor, regarding this isolation part, what are the precautions are taken to prevent the media evaporation?
The main precaution is that the petri
Hello, sir. You're disconnected, I think.
yes, sorry. The plastic bags are used to cover the petri plates to avoid the drying of
Okay.
the agar-agar. Second, keep a beaker containing full water in the incubator so that moisture will be maintained in the incubator.
Okay.
And the drying of agar can be avoided to isolate the thermophilic microorganisms.
Mhm.
Correct?
Okay. Okay. Mhm.
Any other comment?
Brand D'Souza, Kumar Pallavi, Praful Patil.
At least introduce yourself. If you don't want to make a comment, introduce yourself. And again, I'm telling, Microbiologist Society is a topmost organization in India. You can join WhatsApp group of Microbiologist Society. You can give your contact number to Bipran sir. Bipran sir will add you in any one of the group. We are having 40 WhatsApp group of Microbiologist Society. Okay.
If you are a student, then we have also the Telegram group for the students.
Correct, correct. There is a Telegram group to the students. Kumari Pallavi, you are from which place?
Sir, I'm from Greater Noida.
Okay, very nice. Means you are joined from Delhi. Very good.
From which institute you are, Pallavi?
Gautam Buddha University.
So you are pursuing now master degree or?
No,
under-
sir, it's bachelor's in biotechnology.
Aisha is there, Aisha. whether Aisha has introduced Prafulla Patil. I don't think Prafulla Patil and Aisha has introduced.
Also, Jad Sheikh had joined, sir.
Correct. Zayed Sheikh, introduce yourself. Aisha, please introduce. Arvind Kumar is there.
Arvind Kumar, yes.
Yes,
Jad Sheikh, Arvind Kumar.
evening,
Yes,
sir.
Arvind.
Whether you have
Hello.
introduced yourself?
Am I audible, sir?
Yes.
Good evening, sir. Myself, Arvind Kumar. I am working as assistant professor in Maharaja Surajmal Bridge University in Rajasthan, Bharatpur. And
Okay.
currently, yes, sir, I also worked a little bit on thermophiles. So those who are facing, I think it may add something or may be helpful for those. Actually, when I was working on the thermophiles, I used 2% agar while isolating the thermophiles, and that can be sustained on the 60-degree temperature. But you have to keep the keep in the bag with moist cotton, etc. You can use to maintain the humidity properly to avoid the dehydration. So those who are working in there, they can use this. I think this may work for them.
Very true. Very true. Very correct.
So it actually that can be avoided with the dehydration problem. If they are using the plastic bag or something like cage, like something bag or something like container, something where you can keep also the moist cotton along with the petri plates.
Correct. Correct. Correct.
Thank you,
Very
sir.
true.
Thank you, Dr. Arvind Kumar, for sharing your.
Thank you.
Dr.
Thank
Arvind
you.
Kumar is, sir, we are from the same lab, sir.
We are from same lab, yes, sir.
Okay. Very good. Very good.
Thank
Aisha,
you. Uh,
Surjana. Surjana also has joined. Srujana. Srujana, introduce yourself. Prafulla Patil also have not introduced. Zayed Sheikh also have not introduced.
D. Srujana, Kumar Pallavi.
Okay. If
Just
they are not interested, you can conclude. Vipran sir, you can conclude.
I think that Dr. Arvind Kumar put some more insight about this topic. Dr. Arvind Kumar.
Actually, I joined later, so I don't know means the
So
whole
you worked
story.
also on the acid mine, na? Bacteria isolated from the acid mine.
From I isolated the bacteria from some thermophilic Puga Valley.
Mm-hmm.
That was actually tolerant to 65-degree temperature. From there, I was isolated xylase enzyme that is industrially important that degrade the xylem, which is second most abundant polysaccharide on the earth. So that work is actually published now, but as I am not having currently any uh, laboratory such with the equipped laboratory, so I am not able to continue that work. But I am having that culture, thinking to work whenever the facility will be available in my current university where I am working now. So I think this was a very fruitful discussion as per my as I have whatever I have listened up to that, and definitely it will be fruitful for everyone, for the especially for the students also. And I am the member of this from last year 2024 April. Through the Viprans was introduced me in that microbiology society. So thank you so much, Viprans,
Thank you
for introducing me.
for joining us.
So thank you. You please conclude later.
Sure.
Uh,
So, dear participants, just I have shared one feedback form in the chat box. So it will take your 2 or 3 minutes. Just fill out it so that we can issue the e-certificate who had shared their research means experience in this discussion forum. So as we come to the end of this enriching roundtable discussion, I would like to extend my sincere thanks to our All India President, Dr. Arvind Deshmukh sir, and all the participants, research scholars, faculty members, and the scientists for joining this roundtable discussion. Your contribution truly added depth in our discussion and added the meaningful perspectives. So we deeply appreciate your time, effort, and the knowledge you have shared with us today. So thank you everyone for joining the today's session. So just take 1 or 2 minutes and fill out the feedback form I shared in the chat box.
Sure.
Okay.
Thank you, sir.
Thank you, Dr. Lakshman. I think that you can access this feedback form.
Sure. Thank you.
Is it
Yes,
working?
yes, yes, sir.
Is it
Yes.
working? Just fill out it. That will help us to means keep record of the participants and also to means share the certificate to the participants who have shared their research experience in this forum. And I also request everyone to join our session on each Tuesday we organize on the different topics.
Sure.
So only objective is to get connected with each other and to share our research experience to each other so that we can learn together.
Yes, of course. We will join.
So after filling out that form, means feedback form, you can leave the session. So thank you everyone for joining today's session.
Thank you, Dr. Vikrant, and all of you. Thank you so much.
Thank you.
Thank you, sir.
Thank you, Dr. Lokman.
Thank you so much. Good night.
Good night.
Good night, sir.
Good night.